Hey guys, welcome to today's lesson. It is on biotechnology. So the learning goals from QCAA. The purpose of today's lesson is to firstly describe the process of making recombinant DNA. We will discuss this in terms of isolation, insertion, joining of DNA, and the amplification of such. Secondly, to recognize the applications of DNA sequencing to map a species genome. Thirdly, to explain the process of polymerase chain reaction, or PCR, as well as gel electrophoresis. And lastly, to appraise the data. We are actually going to do that one in class, but I will talk to it briefly during this video. So here you can pause the video and maybe just write a summarized version of these goals down in your books before we move on. Cool. All right, let's move on. So recombinant DNA. Here's the learning goal that we just discussed, and I will leave each one up on the slide that we talk through so that you can refer to see if we are meeting that. To begin with, let's say a definition. So this is from the textbook, and essentially recombinant DNA is genetic engineering. It is the deliberate modification of the characteristics of an organism or production of a new organism by inserting or deleting genes into the DNA. So this process involves firstly cutting the DNA of an organism that we want to insert that DNA into. So we can call that the donor, and here in this diagram is the donor DNA. So this process of cutting an organism's DNA uses what we call restriction enzymes. Now you can think of restriction enzymes as a pair of scissors. They are used to cut the DNA or to cleave it. So how do we paste it somewhere? Well, the glue stick equivalent of pasting this DNA are the hydrogen bonds that are found between them already, between the base pairs. You can think of when a cut has been made as those base pairs, those ends, being sticky. But also, DNA ligase is involved in this process. It also joins the phosphate backbones of the DNA pieces together. And so I've got some sticky tape here, and this is interesting because when I pulled it out of my drawer, you can actually see there's this little cockroach just chilling in there. Yeah. So DNA ligase is also used to bring those backbones together. So there is the big picture of how we do it. Let's zoom in a little closer though. So restriction enzymes, what are they and where do they come from? They come from bacteria. This is a bit of background knowledge, but hopefully it helps you in developing your understanding of the entire process. So bacteria have been in this great war with viruses for a long time, whereby viruses invade the bacteria by injecting their DNA material into the DNA of the bacteria. Why? To multiply and to spread. So how do bacteria fight back? Well, firstly, bacteria use this process called methylation. And you should know that term because we spoke about it in the gene expression video. And methylation is the addition of a methyl group to their own DNA, which protects, it coats the bacteria's DNA, thereby leaving the foreign viral DNA exposed. The bacteria then create these restriction enzymes, which their job is to slice, and they slice up the foreign viral DNA material. So the bacteria can then, now that that's cut up, return back to their original shape and purpose. So there's a bit of background information. What does this look like for us and mean for us in molecular biology? Well, we can use these restriction enzymes to cut DNA at specific sites called target genes. So here's a definition of restriction enzymes. They're these enzymes that are there to cleave DNA or to cut. So once we have cut it, we then need to be able to get it to where we want it to go. So how do we do that? Well, we need a vector, something to get it there. And this vector is what we use are called plasmids. So a plasmid is essentially a DNA molecule that is found outside of the chromosome of an organism, but it's typically found in bacterial cells, and they tend to be circular DNA. Essentially, plasmids are used as vectors, used to get this now cut section of DNA to where we want it to go. Okay, so we've got this cut DNA, we've put it into a vector. Then what? We need hydrogen bonds and the DNA ligase to glue the inserted sections of DNA together with the plasmid vector DNA. So here's another little illustration to show this. We've got our DNA fragment from our donor, and we add that to the DNA vector, the plasmid. Then we have our recombinant DNA. How did we get it to stick together? Well, that was the DNA ligase involved in joining those phosphate backbones together, as well as those sticky hydrogen bonds just naturally joining up as well. So now we have this plasmid of the DNA that we want to insert somewhere or into something. So we want to insert this into an organism that can replicate this combined DNA for us, and typically a bacterial cell is used. So we can now place this bacterial cell that has taken up the plasmid onto an agar plate where it will begin cloning itself, and this is called gene cloning. It is also referred to as amplification, that you can see in the learning goal there. So another illustration to show this, we have a target gene. We cut that up using restriction enzymes and we place it into the DNA of a plasmid. This is so that we can create this now recombined plasmid DNA, including our gene. And now we can place that into another organism that can begin replicating that gene that we first wanted. And so it's generally placed into a bacterium. So into, say, E. coli, and placed onto an agar plate where it begins to amplify or clone itself. So that gene that we first cut, why do we do this? Well, it can be used for many different things. One example could be, it could be the gene involved in producing insulin. So we can reproduce that through this whole process to somehow get into a patient so that they can begin producing their own insulin if they're, say, diabetic and have trouble doing that. Recombinant DNA techniques are used in many different fields though, such as in developing vaccines for diseases such as malaria, hepatitis. It's also used in cancer research, plastic surgery, in producing hormones that assist in treating various conditions, as well as in germ cell gene therapy, which involves the replacement of a defective gene with a normal gene. This whole process of recombining DNA was first done in the 70s with the DNA of a frog and a bacteria. So to summarize, you can pause the video here and look over that. But did we meet the learning goal? Well, we wanted to describe the process of making recombinant DNA in terms of isolation of DNA using restriction enzymes, of inserting that into a plasmid vector that would then give us this, but we need to be able to join that up. So DNA ligase is used to join those backbones of DNA together, as well as hydrogen bonds. And we can then place that into a medium that is nurturing for growth and reproduction, or amplification. So that's that process there. Okay, let's continue on. So, firstly, just a little snapshot here. In genomics research, fragments of genomic DNA are inserted into a vector and amplified by the replication in bacterial cells. And that's how we get large amounts of that initial gene that we wanted. Okay. Okay. What does describe mean? It means to give an account of a situation, event, pattern, or process 3 to 4 sentences. So again, lastly, I hope that we've covered that. There we go. DNA sequencing. So the learning goal here, it is again to recognize the applications of DNA sequencing to map a species genome. So there's a few key terms we have to talk about here. But before we do, in order for genetic engineering to even be effective, we need to be able to read the genes of an organism. So before we could do what we just did, we need to be able to know what genes are which. So that's where DNA sequencing is important. It is the process of determining the sequence of nucleotides, that's the A's, the T's, the C's, and the G's, in a piece of DNA. The sequence of nucleotides that constitutes an organism's genetic makeup is called the organism's genome. Keyword there. It's the complete set of nucleotide sequences that make up a species, DNA. And that's found within almost every cell of an organism, of a species. So DNA sequencing maps out an organism's genome, and currently scientists, through this process, we have mapped the genome of about 3,500 different species. The human genome was mapped and completed in 2003 after a many-year international effort that began in 1985, and it was led by this man. Do you recognize him? His name is Dr. Francis Collins, and you may recognize him because we've included a few quotes from this man throughout the course of these videos. He is a Christian, and here's one of the quotes from the very beginning that you may recognize. He said, "The God of the Bible is also the God of the genome. He can be worshipped in the cathedral or in the laboratory. His creation is majestic, awesome, intricate, and beautiful." Since the cracking of the human genome, next generation sequencing techniques are now large-scale approaches that increase the speed and reduce the cost. This took decades to map the human genome before this. This has become a priority in the field of genetics as it opens up the manipulation of disease-causing alleles. And this is already possible. Although this video isn't going to go into the processes of mapping out a species genome, I've included in the bio description of this video a link to a Khan Academy article which talks about the Sanger sequencing method. There's also plenty of videos on YouTube if you would like to check that out, but it's basically these bunch of steps that I've just included a diagram of here anyway. So when we look at the world of genetics today, we are faced with ethical considerations that must be made. A few issues that we must consider include, well, number one, who should have access to our personal genome data? Insurance companies, employers, courts. What implications may this provide to families which have known genetic diseases when it comes to health insurance? For example, who does the genetic material, the data, belong to? Is that to the individual, to the companies, to the insurance, to the government? To what extent should we be able to manipulate genetic disorders? And then where does that lead us in terms of even gene enhancement and gene selection? So there's a few applications there when it comes to mapping the sequence of a species genome. And it can be used in forensics, it can be used in other areas that I just mentioned. But recognize just means to identify or recall particular features of information from knowledge. So I hope that this has been helpful in meeting that. But of course, there's links in the description for you to check out even more. So let's move on. PCR and gel electrophoresis. I wasn't originally going to make a video on this because of Dr. G's videos, but I think it's helpful anyway. So the learning goal is to explain a bit of a high level cognitive verb there, the purpose of PCR and gel electrophoresis. So there are many ways in which we can collect DNA, but the challenge is that it's generally a small or degraded sample. So PCR, defined as a technique that permits the amplification of any short sequence of DNA, can be used to take that small sample and just make it a lot bigger. We can amplify that. So its main goal is to create multiple copies of itself. And this process is called amplification. Taq polymerase, TAQ, we know what polymerase is and what it does, is the enzyme that allows us to use base pairs to build a template strand of DNA. That's like a complementary strand. Primers are also involved in this process of PCR, and they are simply small samples of DNA that are used to target specific regions in our DNA template, where they then attach, and it's kind of like where they attach is where what we are going to be amplifying occurs. So there are 3 distinct steps to this whole process. The first one is called denaturation, the second is annealing, and the third elongation, but in this it's just called replication. So if I just talk briefly through each of those, denaturation, it involves heat. So we heat our sample of DNA up to about 94 to 95 degrees, and that weakens the bonds, these hydrogen bonds between our 2 strands, so that essentially they separate into single strands. We then drop that temperature back down, we don't need it high anymore, so that annealing can occur. And this is where primers join to these single strands of DNA, and we can see that represented there. And lastly, elongation, and this is where polymerase, Taq polymerase, binds to the primers, and it just begins adding these complementary strands. So now we just went from 2 strands to we have double that. And this process happens again and again and again, and you can see from that that we now have a lot more samples of our original DNA. So say we have successfully amplified our DNA sample, the next step is to try and figure out what the DNA actually tells us, and this is the process of gel electrophoresis. In this process, we load our sample of DNA into an agar gel plate, and it's kind of like a sponge in which the DNA moves through, and smaller DNA molecules move further, while larger, slower DNA molecules cluster together at the top. So here's the gel, and we can see the smaller ones, the smaller fragments of DNA, like fragments of base pairs, they just move further. So how do they move? Well, we run a current through the gel. And since DNA is negatively charged, that DNA moves towards the positive electrode down the end here. This allows us to visualize our DNA strands. If we run it across a ladder, say if we put a number of known strand lengths down the side, we can see, oh, well, this one moved this far, so according to our ladder, I can see that that's this many base pairs. We can see that this smaller fragment ran this far, and if I had a ladder there, it would tell me how many base pairs, how big, or what that fragment of DNA molecule is made up of, what base pairs, how many. So essentially, gel electrophoresis gives us an indication of fragments quantity as well as what they are. So the thicker the bands, the greater the quantity of that particular fragment. So that is just an overview of PCR and gel electrophoresis. And we need to be able to explain this. That means make an idea or situation plain or clear. So if you can talk about these 3 stages and what they involve, heating, splitting the strands, adding a primer, adding base pairs, then you're looking good. If you can tell me that there's this current and that we load the samples of DNA into these wells, and then we run that current and the heavier bases stay, but the smaller, lighter fragments of DNA move towards the DNA samples, thereby separating them, then again, you're looking pretty good. So with this video, coupled with Dr. G's videos and your textbook readings, I think you guys should be able to clearly explain the process of PCR and gel electrophoresis. Next, from the learning goals, we have this: to appraise data. So to appraise, what does that mean? Well, we're going to be talking about the pros, the cons, the fors, the against of biotechnology. To appraise means to really think both sides. So it really means to evaluate the worth, the significance or status of something, to judge or consider a text or piece of work. I'm just going to leave it here for now because we're going to do that in class on Friday. Just giving you the heads up. So let's move on, get into a couple of questions. Here we have a multiple choice. The purpose of gel electrophoresis is to A, B, C, or D. Guys, I'll put up a little animation just to help you from Amoeba Sisters, but pause the video and tell me what you get. Okay, so the purpose of gel electrophoresis is to compare DNA binding patterns, eliminate undesirable traits, control gene expression, generate copies. I feel like this is more maybe PCR. It has nothing to do with gene expression, nothing to do with eliminating undesirable traits. So even through the process of elimination, if you had no idea, it's A. Okay. Next question, short response. So here we have it asking, describe the process of making recombinant DNA in terms of insertion of DNA fragments and joining of DNA. This is a written response, and I want you guys to refer to this marking scheme here in your response. So it's 4 marks. We're looking for 4 key points. Let this guide you. Pause the video here and see how you go. Okay, I hope you've had some time to answer that. And hopefully you got something like this. This is just an example. Here's where the marks are. So a restriction enzyme, we didn't say that keyword, it's in your reading. So endonuclease is chosen to digest, that means to cut the selected plasmid at or near the recognition site. So we cut that donor DNA, we cut that plasmid, and then we bring them together. That's where sticky ends come into play. That's the hydrogen bonds between them. DNA ligase is also used to catalyze this reaction, causing those phosphate backbones to come together. So that's a bit of a brief example. But again, follow the marking scheme over this side. Okay, next question. Here we have an experiment was conducted on 4 specific chromosomes regions used in the identification of medicinal plants. The graph shows the success rate for polymerase chain reaction amplification of the 4 loci. The table shows the fragment length in base pairs obtained for each locus. So this is that appraise the data QCAA point. So if we just look at this together and talk through it for a sec before we pause and you answer. Let's look at our table. What are we seeing? We have our 4 loci down on the x-axis here. We then have PCR success rate from 0 to 100 up on the y. And then we have these different values, various success rates depending on the 4 specific regions of chromosomes. But here's our actual values to the side here. So 1, fragment length. Oh, sorry, this is the fragment length over on the side here. So, and the success rate. So depending on the fragment length that we have, one is really small, but then we have higher fragment lengths for these ones. Draw a conclusion about the fragment length, the relationship between this and this. Pause the video here and we'll see how we go. Okay, so here is QCAA's response, and I think you guys would get something pretty similar. It was 2 marks. So you get 1 mark for identifying that fragment lengths of over 35 base pairs are required for PCR success of over 80%. So we could probably call that successful, 80% or more. Then another mark for identifying loci 2, 3, and 4, therefore, can all be used as successful amplification. And there is a exemplar sample response there. There's also some alternative responses that you would still get marks for. And fragment lengths of 85 base pairs have the highest success rate, i.e. almost 100%. Okay. That is it. I hope that this this video has been informing for you, and that it maybe even sparked some ideas when it comes to your assignments, guys. But I thought it would be only fitting to finish with a quote from this man, Dr. Francis Collins, and he says, "One of the greatest tragedies of our time is this impression that has been created that science and religion have to be at war." A cool quote to finish topic 1 on before moving on to our Evo unit. I haven't read this book myself, but a friend told me about it and said it was very good. It's The Language of God, and it was written by this man, the man who cracked the genome. Anyway, I hope this has been helpful. Guys, I will see you in class. God bless.